All right, so we're over the test, so cell structures is done. And with the cell structure, what we did is we focused on the organelles, and we kind of gave you a lot of information on the different types of cells, right? And tomorrow I will go over the exam with you as well in a video. I'm going to make that an assignment. All right, it'll be one of the last assignments of your grade book for this quarter, so it's your last opportunity really to get a super easy grade. And that's for you to watch the video, take notes on it, and then turn in a summary for me, okay? It's gonna be easy. I'm gonna go over the test. I'm gonna send you the test, the video. If you guys spend time doing it, the assignment will be due on Monday or Tuesday of next week, so you have the whole weekend to do. I actually won't be Monday because you're not here Monday, right? So it'll be Tuesday or Wednesday of next week. The assignment will be due. So, this is a really good opportunity for you to get a nice chunk of points that's going to be easy to improve your grade. So please look for that explanation on the test. I will give you your answers back on or tomorrow, all right? And you'll see the difference you'll see the difference in the versions at that point. So most of you, the vast majority of you, know your organelles and you know your cell types. I know that from the test. I know that some of you still have problems with what's an autotroph. All right, so let me retitle this. So trouble areas that we have to focus on is the difference between an autotroph and a heterotroph, right? You guys still have some issues with that. Also, some of you, and more than just a few that did poorly on the test. I have to say, those of you that did poorly on the test, you will be getting remediation. If those that did poorly,
Should it be O after the two?
where?
Between 18 and
Oh yeah, sorry. Let me change the color. I don't even know what I'm doing there. Let me just erase that. Gosh, I hate that. It's hard to write and then auto and then troph. And I apologize because I'm trying to write and speak at the same time. And so, you're saying one word, you're writing another, and you get the letters confused. So those of you that had trouble there are some of you, and it's not a lot, but you are out there that had trouble with the organelles, right? You know who you are. I know who you are. I will pull you out over the next week and give you an assignment to do. That's going to help your grade, number one. It's not for nothing, right? But you need to know what the organelles do. And so I'm going to make sure you know. So that's number one. There will be remediation for that. The other thing you need to understand is that those of you now we're done with the first major set of content, right? This is not Cornell notes or behavior and all that other stuff. This is content that's important for your end of course exam. Do you agree? Mm-hmm. Okay, so now we're talking what you're going to have to do. That section in your blue sheets where it says review all old material, right? Now you have some old material to review. So now I'm going to be looking for not this notebook collection, but the next notebook collection. I'm going to be looking for you, evidence of you reviewing all the organelles. Miss Moore was, and normally I don't like saying names, but I think we should give credit where credit's due. Miss Moore was kind enough to allow me to take some pictures of her notebook. We went over it this morning, and she did an excellent job again. And so I'm going to send you a kind of a close-up look, just a real quick, I should say, outline look at some of the key points of what makes her notebook so good. And one of those is those blue sheets. And some of you are really having trouble with those blue sheets. And honestly, The reason you're having trouble with the blue sheets is because you don't want to do the work behind them. But it's a big part of your grade. It's 10%, right? Projects are 10% of your grade, and that's a big chunk of your project grade. So it's not a full 10%, but it's a chunk of it. All right. So the trouble areas I identified were the autotrophs and the heterotrophs, right? The difference between them. Another trouble area was when you got into details, when you were asked about details of the difference between fungal cells, right? Fungi, protists, plants, animals, cells, and prokaryotes and eukaryotes. When you got to that section, what'd you say? It could be K or C. It depends on how you spell it. I'm sorry. I'm used to reading them both ways, so I'll spell it with a K sometimes, with a C sometimes. It's just, when you read journals, you read it from Britain, and they'll spell it one way. You read it from an American journal, they'll spell it another way. So I'm just used to seeing it both ways. So, anyways, fungi, protists, plants, animals, prokaryote, eukaryote. Okay. When you're looking at these, you got to know the difference. For instance, some people thought that the fungal cell was an autotroph. They're not. They're heterotrophs, right? Some people thought protists were multicellular. They're not. They're single cellular. Some people thought that, confused. In other words, some people confused plant cells and fungal cells. There were a lot of cell types, so I could understand that some people could get some stuff confused, but you can't let this go on. This is an issue that you have to address now, because you can't just wait till May and find out, I got that question wrong on the end of course because I didn't correct it. So we will go over it. I have some, when I go over the test, I want you to take notes on that and summarize those notes and pay extra attention to these differences. Prokaryotes are not only single cell, but they don't have a nucleus. Eukaryotes can be any one of these. A eukaryote can be a plant, an animal, a fungi, or a protist. A prokaryote can only be bacteria or an archaebacteria. Archaebacteria. It's the only way I can spell it right. So I remember archaebacteria. All right? Are we good with this? So these are the trouble areas we will be dealing with as a class. Most people got organelles and types of cells overall right. Almost everybody confused one part of this or another. And when I go over that part of the exam, you'll see what I mean, how tricky some of these can be. So everybody should be happy with the results. I think you guys learned a lot, so we can move on. Yes?
Yep.
Okay, so let's move on. So one of the things that we have to talk about a few things in our next goal, because you want to talk about what we're talking about this week for your blue sheets. This week we're going to focus on the membrane, which means overall we're going to be discussing in the membrane phospholipids, it's a word you better get used to reading. We're going to be talking about things called integral membrane proteins. We're going to be talking about channel proteins. And I shouldn't say just this week. This week, let's just say this week is what we're talking about here, okay? And channel proteins, and we'll be talking about also transport across the proteins, or across the membrane.
What does that say that I want to get out?
Integral.
Oh,
Integral Transport across the membrane. Transport across the membrane, which means we'll be talking about what crosses the membrane. These are the big ideas. If you want to know what you're going to be tested on, I'm literally listing it for you right now. Right? So when we're going over this in a minute, and when we continue to go over it, and when you get a project on Thursday and Friday, and then next week, over the weekend and such, you know what you have to know. You should know what index cards you have to make, because it should be easy. You saw from the chemistry of life stuff, you saw how some of you didn't memorize those shapes, and it came to bite you, didn't it? It came back to bite you. Don't make that mistake now. You know, I'm telling you what you need to know. Start learning it a little every day. Don't wait to the end. Don't try to force your brain to try to get everything in at once. It might work in the short run, but it won't work in the long run. Transport across the membrane. What crosses easily? And here we're going to be talking about one, polar and nonpolar is going to be a big deal. And what's ionic versus not. And do you see how things build? Do you see how everything we covered is coming back? And now you have to use these definitions to understand what the heck is going on with transporting across the membrane. Some of you that didn't learn this and early on, or this early on, now you're saying, oh my God, there's so much. What are you talking about? It's not so much, because you should have already learned this, right? We already learned about lipids. Now you're just going to learn about phospholipids. But if you didn't learn about lipids before, now you got double to learn, don't you? So please, if you're feeling overwhelmed, learn a lesson. Learn it when I'm teaching it to you. Pay attention. Take it seriously, because it's all integrated. I don't teach something to you just for fun. It's necessary so that you understand the big picture. All right, so what crosses easily and what doesn't, right? Then the next thing you have to figure out is something called diffusion. Now we're working into next week. Now we're talking about next week now. So this is week 1. This is this week here. Now we're going to talk about next week. Diffusion. Osmosis. Active transport. And passive transport. So there's going to be a lot of discussion. It's going to take a week. You're going to have a couple labs. You'll have 2 labs here. What's this? Hold on a second. I have to pause. All right, so diffusion, osmosis, active transport, passive transport is all we're going to cover next week. So in order for you, this is what I keep telling you. For you to understand what's coming next, you have to understand what came before. If you don't, it's going to be that much harder. And we all know what that feels like when you're confused. So please, let's keep it in line. All right, let's move on. Let's talk about phospholipids. All right, what are lipids? All right, so that's good. Fats, I heard fats. Oils, right? Can you describe a lipid for me? This is all review. This should all be review. There's unsaturated. Yeah, okay, I'll put that word down. Unsaturated. Unsaturated. And yes, that saturated, unsaturated does have to do with the hydrogens, number of hydrogen bonds, or a number of bonds to hydrogen from carbon. So what are they in general? There's an idea that was, when we went over lipids, it was a really big idea. It was on the test, on the chemistry of life test. Every question I put on a test, I know it's coming later, so that's why I put it on there. So you have this description of lipids that's always true. Remember, anybody? Okay, so I'll go ahead and I'll review it now. That's why we're doing this. All right, so lipids are long chains of what? Carbon. Mostly carbon and hydrogen. And so it's, let me make sure you understand. We're talking about mostly carbon and hydrogen. Those are lipids. Lipids are mostly carbon and hydrogen. Remember that? What do we know about carbohydrates? What are they? Carbon, hydrogen, oxygen. You see, these lipids are carbon and hydrogen. And we usually don't write the hydrogens when we draw the structures. Sometimes we do, sometimes we don't. But each one of these lines is generally attached to hydrogen. Remember that? So is this, somebody close the door. Is this saturated or unsaturated? Saturated, because you have all these hydrogens on there. So that's going to become important when we talk about the membrane. So that's why I'm going over it. It turns out that on this end, what's on this end is important. Because, see, this whole section here is what we call what? What do we call that? So long carbon-hydrogen bonds, what are they? Are they polar or nonpolar? You're right, it is a lipid. Somebody said it, that's true. But what is it, polar or nonpolar? Oh, it's too bad. A lot of review has to go on. This is nonpolar. It's an oil, it's nonpolar. Who said that? Nice. Don't say that, your name. I don't want your name out here. So yeah, that's right. She's absolutely right. It's nonpolar. And remember that whole chromatography lab. Why did we do the chromatography lab? To go over polar and nonpolar. Why was that important? Because I knew it was coming, right? So this is nonpolar. So that's going to be dissolved by what? The same thing, because like dissolves like. Do you remember that? Like dissolves like. Really important. Really important. Are we all good with that so far? But you see this thing here, whatever's at the end is going to make this, could make this molecule very, very, very different. Whatever's at the end of this molecule can make it very different. In what way could it make it different? How could what's at the end in that question mark, what could make it different? The molecule is nonpolar right now. What could we add to the end of it, to the end of this chain of carbons to make it nonpolar, to keep it nonpolar? I heard oxygen to make it polar. Is that what we're thinking? Yeah, adding an atom like oxygen would help to make it very nonpolar. You're very right on that. Isn't that interesting? That was very good, because remember oxygen and nitrogen, they form nitrogen bonds, or hydrogen bonds, when we attach a hydrogen to those two atoms, because we didn't go into the details because it's not chemistry. But it makes that uneven sharing of electrons, right? Is everybody good with that? All right. So oxygen's one. That's very good. What else might we be able to add? A nitrogen. That's right. That's right. So we could add all kinds of things to it, because we could add anything to it. This is chemistry, right? So we have 100 and some odd elements to choose from to put onto the end of this, and we can make this lipid into anything. If we add a carbohydrate, what would we call it? It would be a carbohydrate attached to a lipid, right? If I had a protein, it's a protein attached to a lipid, right? So I could add anything I want to it. Some things are more stable than others. So let's look at what our bodies do. What do bodies? Now I want to say our bodies. I got to say all cells on the planet. And now today we understand when I say all cells, there are a lot of different cells, aren't there? We got animal cells, plant cells, protists, fungi, and prokaryotes, which include bacteria and archaebacteria. All those different kinds of cells, plus all the different kind of subtypes, right? So there's a lot of different cells, and they all do the same thing. Isn't that interesting? Because if you think if it was going to be random, if we weren't all related, this is kind of the evolution link here. This is something to think about when we're talking about evolution. Evolution says we all came to the same thing. What's some of the evidence that we all came from one of the same living creatures? Some of that evidence is that all our structures are the same. All our cells, cellular structures are the same. Or at least very similar, I should say. When we look at cellular structure, there are slight differences, but the striking similarities is hard to ignore. Evolution explains the similarities. So I'm going to draw out, it turns out that we have this thing called glycerol, right? And it sounds like a crazy name, doesn't it?
Like a cleaning tool.
Like a cleaning tool?
Like a cleaning.
Is it? That's a good question. See, I love the questions I'm hearing. I'm glad that. And let me. Yeah, I'm gonna draw it this way for a reason, but I want to move it over. So it sounds like a cleaning tool. I heard. And somebody asked, "Is it a carbohydrate?" And why did he ask if it was a carbohydrate? Anybody know? Yeah, but what? There's another clue, and that's that all. OL. So the molecule's called glycerol. Very important little molecule. Have you ever heard of the word alcohol? Yes. Oh, yeah. Yeah, glycerol. We knew if it was a sugar, if it was a carbohydrate, if it was a sugar, what would we expect? Ose, right? We'd expect it to end in ose. So it's not quite a sugar. But or at least it's an alcohol, right? It's very similar to sugar. A little different, the sugars that we've looked at anyways. So glycerol is just this. It's real simple. It's a simple little molecule, but as simple as it is, wow, is it powerful. It's the beginning of a lot of a lot of molecules. It's actually a very important molecule. It's the beginning of all lipids. All right, so you have these 3 carbons. It's just 3 carbons long, and then they're all attached to an oxygen and a hydrogen. Remember, each oxygen needs how many bonds? Anybody remember that?
4.
2. Each carbon needs what? And you see, good. I'm glad you remembered the number 4. That was important. And I'm very impressed that you guys are able to learn all this without knowing a lot of chemistry, right? Like if we spent a lot of time doing chemistry and you got that wrong, I'd be very, very upset. But you got that wrong, but you were close and you don't know chemistry, right? You haven't been taught chemistry, have you?
No.
No. Yeah, a little chemistry, a little biochemistry, but not a great deal of chemistry. So I'm very proud of you that you know that number 4. But that 4 is for carbon. Oxygen is 2. Question. Well, that's a good question. Why does one molecule end with 3? And in the case of glucose, why do we get 6 carbons, right? And why 5 carbons in the case of ribose? It has to do with the chemistry. It has to do with the energy, amount of free energy available. It has to do with the enzymes that are involved in making it. And so the answer gets complicated. But just to say that different chemical reactions produce different structures. And this particular structure is glycerol, and it produces this 3-carbon molecule. Very important. Well, it turns out that if we take, and this is what happens, all right, we're going to do what's called dehydration synthesis. Remember I promised you that we were coming back to that? Do you remember that or not? Some people do, some people don't. Okay. So we're going to go ahead and try to show you a dehydration synthesis. So you got this big carbon chain. This carbon, hold on a second. So what happens is that you got this chain of carbons, and the length of the carbons can be different, all right? So I'm not going to get in too much detail on that. Here's your hydrogen, right? See how long it is? It's actually longer than this, but I don't want to draw any more carbons. Can I be lazy? Is that okay with you?
No.
Oh, you want me to draw out the whole thing? That means you have to draw out the whole thing. This is close to how many? So you got this carbon-hydrogen long chain, right? So this part here is what? Hydrogen, carbon, that's right. But is it polar or non-polar? Non-polar. If you weren't able to answer that quickly, what does that tell you? You need to make sure you know the difference between polar and non-polar, right? So that's one of the keys. I'm telling you, it's coming on the next test, so you know it already. Now this last carbon's not going to be attached to hydrogen like the rest of them are. This last carbon is going to be attached to double bonded oxygen. And then can this oxygen bond to anything else? No, because it already has two. Do you see that? Oxygen can only make two bonds, and there it is. There they have it. Each of these carbons can make four. You see it? 1, 2, 3, 4. So each of these carbons are filled. Each hydrogen can only make one. So everybody's happy so far, except for this poor carbon only has how many?
2.
1, 2, 3.
Oh, 3.
3, right? So we need one more bond. Well, it turns out that one's bonded to OH. OH or HO. All right? We call it OH because it's hydrogen. Now that's a saturated one, right? But we can make the same thing. Can you see us making another one? So C, 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 and C. So those carbons, there's some hydrogens here. Remember, you could do this by now, can't you? You can make sure that there's 4 bonds to each one, except that there's a double bond here. So let me undo that. Let me erase this. Why does it do that? It gives me the option. All right, so let me say this. And when you have a double bond, what happens is it has to do with a lot of stuff that we don't have time to talk about, okay? But when you have a double bond, that's going to twist the molecule. It's going to turn it. So when we turn the molecule, let me erase this for a second again. What's going to happen is this thing's going to turn like this. Maybe not so severely, but it's just to make a point. And what you're going to get is this. And so when you have that double bond, well, it's still a lipid. That's good. I'm glad to hear someone say it's a lipid. So you know that this is a carbon here, and that's a hydrogen. And this one's unsaturated, isn't it? That's right. When it's a double bond, it's unsaturated. But the key for us, for health reasons, the unsaturated is better for you, right? But the saturated is probably okay, but not as good as an unsaturated. The one we don't want is a trans, and we talked a little bit about that. But when we're talking about the membrane, it's important to note that twist, this area here that turns, becomes very important for constructing the membrane. It creates fluidity. You want your membranes to be fluid. You want them to be relaxed. You don't want them to be stiff. If your membranes are stiff, they can't adjust well to the environment. And when it gets cold, what do you think happens to fats?
They freeze.
They become harder. You've seen fat in the pan. When you fried up a steak, have you ever fried up a steak or bacon or any meat that's fatty? You take your steak out, you eat it, oh, you're done. And mom says, go wash the dishes. And you go back to that pan, what do you see? Yellow chunks of fat. It used to be liquid, now it's solid. Why? Because it cooled down. You don't want your membranes to turn into that, do you? You wouldn't be able to work. It wouldn't work for you, right? You don't want your membranes to be that stiff fat like butter, right? You want your membranes to be more fluid. So this allows them to be more fluid. It allows them to move around. So some unsaturated fats in your membrane are a good thing. All right. All right. So of course, the end piece here is also again connected to a carbon. This connected double bonded. Oh, I did it. Double bonded oxygen to an OH. Do you see how that goes? Is everybody okay with what we're seeing here? Is there anybody that's confused about something? You know, now's the time to say, hey, you know what? I don't see that. I don't understand what you're saying. You know what? It's not, don't wait till later to tell me that, oh gosh, I didn't understand that. I don't know what's going on here. We don't want to do that, do we? Do we want to wait till later to say that?
The HO means hydrogen, oxygen.
The OH is an oxygen and a hydrogen group. We call it hydroxide. But the OH group, there's a lot of OHs, right? If you remember carbohydrates, we have a lot of OHs. OH is a real major combination of atoms that we see in biomolecules. And that you'll see why in a minute. You'll see why OH is important. Hey, what's water made out of?
H2O.
H2O. Let's write that down. H2O, and that's an oxygen with two hydrogens. H2O. OH. H2O. Are we seeing any similarity? OH. 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 So if we're going to combine these molecules, right? If we're going to combine these molecules, we have to get rid of some bonds, right? Do you agree?
Yes.
And remember, chemical reactions are what? When we take some atoms and move them out and replace what's bonded to what? So if we're going to take these fats, we're going to add them to this thing here called glycerol. So this is important. How are we going to do that? We have to get rid of some of these atoms, don't we? Because they don't fit together. They're full. All that bonds are full. We got to get rid of some bonds. We got to get rid of some atoms. So what we're going to do, and let me pause this again. Yeah. So what we're going to do, watch what I do. Watch my circle. I'm going to use a pen to circle what's going to happen. Are you ready? Let me draw out, I'm going to draw out the oxygens in red. The oxygens that are not going to turn into water. They're going to turn into red. The blue oxygens are going to be part of what? Water, right? Going to be part of water. You see it? There's an oxygen in water. Do you see that?
Yeah.
I mean, I need you all to see that. Please just don't say yeah, and then people get surprised. What is it? There's an oxygen there. And that's this oxygen here. Any one of these will turn into water. Okay? Because they're gonna combine with what? What are they gonna combine with? The hydrogen. They're gonna take this. We got to get these out. It has to do with chemical reactions. It has to do with chemical potential energy. It's gonna have to do with all this stuff that we don't really want to get into. I'm just telling you what happens. We're not getting into whys, okay? There are really good reasons why this happens. We're not going to talk about it because it would take a long time. Are we clear? So what happens is that this hydrogen here and that hydrogen there, do you see what's happening? They're going to leave. And this hydrogen is going to leave. And I hear people talking. I hope you're listening. You can't do both. So what's going to happen? What's going to leave in this molecule, see if it moves here. What's going to leave here is what? What's going to be leaving? To what? To H, twos, and an O, right? Two waters. Do you see that? Yeah, so you got the hydrogen here, another hydrogen here, and the oxygen. H2O. H2O. They're leaving. That's why we call this what? Dehydration. Why do we call it dehydration? Come on, guys. We can break this down. That's right. We're taking a water away. Dehydration. Dehydration means we're taking, D means not, hydration, water, right? Dehydration synthesis. Why do we call it synthesis? What does synthesis mean? Synthesis means making, to make. To make. Dehydration, making. Does that make sense? What are we making? We're making water, but we're also putting together the other two molecules. We're making a new molecule. Do you see that? You see how we're putting things together? This is a putting together reaction, right? Do you agree? Any putting together reaction, we call it synthesis. All right, so we're taking this and we're taking this, we're putting it together. You see that? We're taking these two fats, these two lipids, with the nonpolar sections, and we're connecting it to this thing. Everybody cool with that?
Yes.
All right. So that's why we call it dehydration synthesis. Another word for this, a kind of more general term, is condensation reaction. Somebody used it earlier. You might see both or either. Condensation. Why does condensation seem like it makes sense as well? What is condensation? Water making, right? So that makes sense too. So water's formed when we put these things, when we make a phospholipid, we make water because water's coming out of the reaction. Okay, so then, but what happens to these? What do these look like? Well, let's draw it. Can I go up? These two. What are these two? What is this? After we get rid of the water, what does all this look like? All this is going to become one big molecule, right? One molecule. So let's take it. Can I make it smaller now? Everybody good? All right, let me make it smaller. And so let's look at what it looks like now. This is before. Now let's look at after, right? After we put it together. There's a carbon. You agree? Hydrogen. Please help me out. I'm lost. What goes over here? What's it connected to?
H.
The H is gone. It can't be the O and the OH. Let's look over it.
So it's a carbon?
The O, the H, and the H are gone. It's an O. It's an oxygen.
Oh.
It's connected to an oxygen,
And the hydrogen.
and then it's connected to a carbon. And you got it double bonded to what? An oxygen. So you got it. You see it, right? You see it. 3. How many are there? 1, 2, 3, 4, 5.
6.
3, 4, 5.
6.
Is there 6? 1,
no,
2,
no,
3. No, 5. 5 in the original drawing. I think there is, and it usually 3, 6 or 7 is fine too. Whatever. I'm just drawing 5 here, okay? And I just want to keep the number constant. But how many? It depends on the fat, right? It depends on the lipid as far as how many carbons. But you get the point, right? There's a long chain of carbons here. And remember, what's this part here? I'm going to keep saying it. What's this part?
Nonpolar.
Nonpolar, right? I'm going to keep drawing it. So of course, that's not all the glycerol molecule is, right? We got another carbon here. And what's that connected to? Another oxygen, right? So we got it connected to another oxygen. And you see why I color-coded it so you can see where everything's going. So that's connected to a carbon that has what? Another oxygen. And then it's connected to another carbon that has a hydrogen. But then that's what? What's that one connected to? A carbon that has what? A carbon that has a double bond. That's right. So that carbon has a double bond that turns it, doesn't it? That turns it to another carbon. It's double bonded to another carbon. And we'll make this cis. And so there we have our cis double bonded. But we're not done with this glycerol molecule. We got what? What do we got left?
We got another carbon,
We got another carbon here.
but you have an OH.
That's right. That one still has its OH, doesn't it?
Yeah.
Let me draw it in blue. That one still has its OH. So we could still build something with this one, can't we? The others are pretty much done. I mean, we could take them apart and put them back together again. That happens in chemical reactions. When you eat fat, that's what you do. You break it down. We do the opposite of dehydration synthesis. We call it hydrolysis, but we'll talk about that tomorrow, okay? So dehydration synthesis is when we make something, we just made something. So this is cool, but it's still not a phospholipid. It's still not a phospholipid. So right now we're close to what a phospholipid is, but not quite. Now if we added another fat here, we would call that a triglyceride. Glyceride because of the glycerol molecule. Triglycerides, when you go to your doctor and they do a blood test, those of you that want to be doctors and nurses in the near future, when you go, your parents go, they'll tell you how high your triglycerides are, right? So you don't want high triglycerides in your blood because that means your blood's full of a lot of fats. You don't want that, right?
It's a high number.
We're not going to get into that. But just to know that when we're talking about triglycerides, we have another fat on there. But if we add, what happens if we had, instead of a fat, what if we add another molecule that you've all heard of that is in DNA? There's another molecule that we've all heard of that's in DNA. Phosphate, excellent. That's exactly right. We could add a molecule called phosphate. If instead of this glycerol, these long chain fatty acids, we added something called phosphate, then, and it's not just phosphate by itself, I wish it was, but there's a little bit of a head to it. But I'm not going to draw it, if you don't mind, because it's going to be a little bit, it might be a little overwhelming. But what if instead of that, we have this thing that has a P, I'll just draw it off on the side, a P, and then an O, and then an O, and then an O, maybe there's an H here or something, OH, and then this is connected to a carbon and a hydrogen, and another, let me move this down, and this is connected to another carbon and a hydrogen and a nitrogen and a OH, and just all kinds of stuff on here, all right? Can we just say stuff? Can I say stuff?
No.
The only, I hate you. Okay, no, I'm kidding. Here we have an OH. So if we take this H, the reason I wanted to draw this part is number one, everybody, you know what, I messed up.
What's that?
That's a helicopter?
No, that right there. At the top.
Why is it not?
With the little cross at the top.
Wow, it's a plus sign. It's a plus sign. It's a whole bunch of atoms there I don't want to get into. I'm just kind of trying to let you know that there's more stuff there, okay? There's more stuff there. I messed up a little bit. For some reason, this is not letting me erase. I don't know why. Oh, there it is. So what we got to do is there has to be another oxygen. Because remember, the phosphate we learned looked like this, right? And that's it. And by the way, this is how we put DNA molecules together, DNA nucleotides together. That's why you have a phosphate sugar backbone, because it's the same thing. It's dehydration synthesis. Use dehydration synthesis to put most of your polymers in your body together. Now, a fat is not a polymer, but we still use dehydration synthesis. Every time you make something in your body, and all living things on the planet, including bacteria, this is the kind of process they use. This is the kind of process we all use. All life on Earth that has cells.
So that you drew a phosphate?
This part, right? Let me draw it out. This part here is a phosphate. All right. This part up here is something. It's just a head. We'll just call it something else. It just sits there. I don't want to talk about it.
The one that you just drew at the top.
Where did I draw what at the top?
At the top of the screen.
Oh yeah, this is a phosphate. Yeah, I was confused. I wasn't sure what you're talking. Yeah, this is a phosphate. So every time you look at a DNA molecule, you see that. Remember, we said when we did DNA, we said, look, instead of doing that, we're going to draw it like this. Do you remember that? The P with the circle around it. We'll call that. But when we drew that P with the circle around it, we were talking about this thing here. A phosphorus atom with 4 oxygen atoms attached. I don't know why you think it looks like a D. It's clearly a P. All right. I don't know. All right, so I just want to try to focus. We're running out of time. We only have a few minutes left. I want to make sure you get this before we go. So this phosphate head, what do you notice about this thing? You have the hydrogen here. What do you think is going to happen?
It's going to create
It's going to come and snatch this out, isn't it?
Yeah,
And create what?
water.
It create water. That's right. So water comes out again. It's the same thing as always. It keeps happening. H2O. Dehydration synthesis, right? Which one?
Condensation.
Condensation, dehydration, same thing. Right, so it's just because you're making water. That's why we call it the same thing. So, does everybody get that so far?
Yeah.
So when we sit there and we do this stuff, dang it. Huh, interesting. Interesting. Give me a second. All right, so again, this is dehydration synthesis again, but what do we have now? Now we added something else to it, right? Now we got this big long molecule. Let me go ahead and draw it out, and then I'm going to give you the real simple way you're going to have to look at a phospholipid. See, you don't have to, I'm not going to ask you to draw all this out. But I am going to ask you to be able to recognize when you see a phospholipid tomorrow, when you're constructing a membrane, that you remember it's all this. It's not just a little squiggly line on a piece of paper. All right? It's so when we're looking at a glycerol molecule, and by the way, you'll see glycerol again when we talk about photosynthesis. But there's this glycerol molecule, and I'm going to draw it differently. Is it okay with you? Will it confuse you if I draw it horizontally instead of vertically? I hope not, because it's... So I'm going to go ahead and draw it vertically now instead of horizontally, rather. Here's a carbon, there's a carbon, there's a carbon. Yes? That other little thing, this thing here, I don't have time. It's not like you have to know it, but I know some people are asking about it. It's choline. All right, but you have choline, you have glycerol, and you have phosphate, and then you have lipids. So the whole thing's a phospholipid, and that's what we're constructing. So when you're looking at this, and you're looking at this chain, we're going to go ahead and draw 1, 2, 3, 4, 5. Let's just go ahead and put 6, even though we did 5 before. All in a row, right? And then all these are hydrogens. There's an oxygen in between, so that was badly done on my part. So there's an oxygen, and then, right? Do you agree? And you guys aren't helping me, so that's probably why I'm messing up.
What do you know when it's double bonded?
It's just what it is.
Oh.
There's no way for you to tell what's going to be connected here. It has. So you see, let's go ahead and put the double bond. You just, in this case, it's whatever it turns out it is. It depends on how it was made, what it's made from, what conditions was it made at. Made under, okay? There's a lot of talking going on. I hope it's about what's going on, because you're going to be tested on this. You're not going to be tested on drawing it, but you're going to be tested on the ideas. And this is where you guys are always like, I didn't know it was going to be on the test. Oh, woe is me.
So
And I tend not to have too much pity for you, because really, you should know. So there it is. That's the first end, right? And then, of course, here I'm going to have what? Another oxygen. Should I draw it red like we did before so you see the connection? All right, so there's another carbon. There's another oxygen. There's a carbon. Let's make this one. I know it's time to go. Let me finish drawing it before you go, if you don't mind. Just because I don't want you to have to spend any time at home doing this. Of course, you could decide to go but then you would disrupt everyone else. So that's the middle part, right? And then the last part is there's no long chain carbons now. See, this area here is all nonpolar, isn't it? This is all nonpolar. Long chains of carbon and hydrogen. But on the end, you got this thing that's sticking up here. That instead of drawing out this whole big long structure, are you okay with me doing this? And putting a P here with a circle? A P with a circle is going to be all that stuff I drew before. So now, I heard people tell me it's 9:57. Did you all hear me say that I'd rather you just wait a minute? I know it's time. I know you got to go to advisory. I get that. But what if I draw this a little differently? What if I give you an easy way to draw it? Do you think you can remember the easy way of drawing it? Yes. Even though some of you aren't paying attention to me at all.
Yes.
What if I tell you to draw it this way? Take this whole thing and call this phosphate All right, phosphate, choline, everything all together. Let's call this the polar now. Why? Because what is phosphate? Phosphate is polar.
Polar what now?
The polar head.
Oh.
And I tell you that instead of drawing all these oxygens and carbons, all these double bonds or anything, what if I drew this? Call that a hydrophobic tail. And the other one, I'm going to put, what's this? Why does this one come off to the side like that? That's the double bonded one. Excellent. That's the one that twists out like this. This is the one that's unsaturated. This one here is the phosphate head, right? Phosphate head. We know the phosphate is more than just phosphate, right? I just drew it for you. We know now that we made it through dehydration synthesis, right? But this is what we call, this part here is a lipid. Do you agree? It's nonpolar. I drew it in yellow so everybody knows it's nonpolar. There's a double bond here. This is unsaturated, this is saturated, but they're both nonpolar. And instead of the polar head and the nonpolar what? What would you call these? Nonpolar what? Can I call it a tail?
Yes.
All right, so nonpolar tails. That's it. That's to be what you need for tomorrow's activity. Good luck. Thank you for your patience. So you have a question, go ahead.
Okay, so when there's a double bond, okay, so when a carbon
Here,
is...
point using this so the people at home can know what you're talking about.
Okay, so when this carbon here is double bonded and then is bonded with another carbon, could it only be bonded with one other?
That's right. That's right. So this is badly drawn, but I was rushing. So really, this is another hydrogen here, right? And this one only has two, so it really needs two more hydrogens here, right? This one has all four. 1, 2, 3, 4, so that's fine. This option is 2, that's fine. This has 1, 2, 3, 4, right? So it can only be bonded to 2 other things.
Okay.
And because it's bonded to 2 bonds out. Now this is badly drawn as well. I was rushing. It should have been a carbon, right? Not an oxygen. So actually, let me see if, I don't know if this will let me erase it, but if I can. So this really should be a carbon. I do it too quickly.
So it's double bonded with a carbon.
Right, so then now what we have is carbon, 1, 2, 3, 4, carbon, 1, 2, 3. And so what does this carbon need?
The hydrogen?
A hydrogen on it, yeah. And then when we look here, 1, 2, 3, 4, right, all 4. That's a hydrogen, it's badly drawn then. 1, 2, 3, 4.
Oh.
So yeah, the good point, 1, 2, 3, 4. See how these are 4, 4. All right.
So it has to be bonded with 4, or it's just...
Carbon,
it always
I'm going to
has
say always.
Okay.
In chemistry, it gets more complicated, okay? But what we want to say, when you look at a biomolecule, carbon's always going to have 4 bonds on it, all the way down. So if there's not 4 bonds, there's something wrong, or it's something that's a special condition, right? But we're talking about carbon having 4 bonds, yeah.
Okay.
And of course, you have here phosphate, and then you have your nonpolar tail. So now you know what this is going to do. And tomorrow we'll talk about what happens when you get a bunch of these together. All right, when you get a bunch of these together, can you imagine what's going to happen? Just to give you a little advance notice of it. What do you think happens when you get a phosphate head, a polar head, with 2 nonpolar tails, let's say, and you get a bunch of these together? What do you think is going to happen? If you get another one of these, you get a bunch of these in water. They're going to be floating in water. What do you think they're going to do? What are these tails going to do to each other? What are the heads going to do to each other? Remember, like dissolves like. This side is what? What are these?
That's saturated.
Saturated or unsaturated, yeah. But what else are they?
Nonpolar tails.
That's right, nonpolar tails. So like dissolves like, what's going to happen? The polar will dissolve the polar. So what's going to happen is you're going to get the polar tails all interacting with each other. And then Give me a second. So what's going to happen is that you're going to get all the not. If this is in water, remember this is all in water. There's water all. Oops. You got water all in here, right? All in between them, and in between all the water's a small molecule. So what's going to happen is you're going to get the hydrophilic heads. Hydrophilic, right? Hydrophilic. So let me write that out for you. Hydrophilic, which means water loving, which also means actually polar. And hydrophobic means what? What does phobic mean? You have a phobia. What is it?
Like a phobia, like a fear? Like
A fear. Yeah.
Oh, okay.
So hydrophobic means that water, they're afraid of water, right? Philic means it loves water, right? These are ancient terms. So hydrophilic means they love water. So these phosphate heads, they're polar. They love water. They're going to be dissolved by water. So they'll dissolving meaning they associate together. So these phosphates will associate with the water. The nonpolar tails will associate with each other. They kick water out. So we create the first, what we call, this is called a micelle. It's the beginning of what a fat is, or what a lipid bilayer is. So it's not quite a cell. It's not a cell. It's not a cell membrane yet. But a micelle is just this little round interacting ball.
Like
and,
that.
Yeah, like that. And I always do MY, but it's MICELL. So it's a micelle, and all a micelle is, you get the hydrophilic heads pointing towards the water and the hydrophobic tails pointing away from the water and towards each other. And tomorrow we'll look at what from a micelle we'll go into how do you make a bilipid membrane.
So the tails are facing from the water.
Away from the water.
And the phosphate heads
Are pointing towards the water.
Towards the water, okay.
Yeah, and that's the key. All right? Mm-hmm. All right.